Welcome, everyone. It's my great pleasure to introduce Alistair Boettiger. And today, I'm Pallav Kasuri. I'm a faculty at the Salk Institute and also have an adjunct appointment here. But my most important credential is that I was fortunate enough to be colleagues with Alistair for a couple of years at Harvard Chemistry in Xiaowei Zhuang's lab, where we both did our postdocs. And let me take a step back and introduce Alistair and why I think he's so spectacular, both as a person as a scientist. So Alistair started with humble beginnings at Princeton as an undergrad, where he studied both physics, a little bit of computer science, and also biology. And then went to do his PhD at Berkeley with Mike Levine, studying all different flavors of enhancers, of which they seem to be a lot these days. And did some phenomenal work on development there. And after that, did his postdoc in Xiaowei Zhuang's lab at Harvard Chemistry. And that's from where I know Alistair, and he's been a fantastic mentor to me and to Bogdan, who is in the audience, and didn't overlap with Meng, who is here today, but we have there's a lot of culture in the lab that stemmed from the innovations that Alistair contributed. And in particular, I want to point out one innovation that might not have gotten so much attention, but I think is really profound for the field of spatial transcriptomics and digital biology in general. And so when Alistair was in the lab was really developing this method called MERFISH that we know is really powerful way, and there's many different versions and flavors of this method where you can get a multiplex readout of features in a cell. So initially it was transcriptome. Some people like Bogdan have pioneered other uses of this method, and I think the fundamental concept underlying all of this is to turn analog signals, fluorescent signals, into a digital readout. And that concept is really powerful because then you can use computer science methods to decode, to error correct, to check for errors, and even get a quantitative metric of your error rates that underlie all of our measurements. And so I was trained in more analog biology. Actually, I was a physicist, but then I trained in analog biology. I was always so frustrated that these fluorescent signals are ratiometric and they're semi-quantitative. But by really getting signals that are super bright, you can set a robust threshold. So just like in digital signal processing, it's either on or off. And once you can make that call, you can start to incorporate these error correction modes. And Alistair was really the person who brought this idea to the lab that, wait, why don't we borrow some of these methods like Hamming codes and error correction from the field of computer science and apply this to our now digital biology readouts? And that became revolutionary. In my mind, turning these analog signals into digital signal processing and essentially computer science has been groundbreaking. And so Alistair did that sort of as a side project. His main project was actually looking at the conformation of the genome. And after he started his own lab, I think you've done a bit of everything, and I'm really excited to hear some of the latest work in live cell imaging. But I also would be remiss if I didn't mention some of your fantastic awards that you won. You're a Searle Scholar, you're a Cobley Fellow, a Packard Fellowship. You won the New Innovator DP2 Award from NIH, and we're both fellow Beckman Young Investigators. Many more awards to come and some fantastic research on the menu. Take it away, Alistair.
Thank you, Paolo, for the very flattering introduction. And I'm incredibly grateful to the many colleagues that I got to learn from in my scientific journey and who I followed into various adventures, into following things where you don't know what you're doing. Works okay in the lab. I did learn following Pallav down some adventurous ski slopes was beyond my capabilities for what he can do. So be careful if following the ski master here as well. But with that, I will dive in. What I'd like to tell you today is about some new directions we're doing in the lab. We're just beginning to talk about it. It's not written up anywhere. We haven't submitted anywhere. So I'd really like feedback. And I think this is a lot more fun and engaging, and I'll get more feedback. If you stop me mid-throw and let's talk about things as I go along, and it will be more interactive. Then we also won't finish 20 minutes early. So with that, the guiding questions, I think, for my team and what has inspired me since I was a graduate student is really understanding genomes, in what sense this is a developmental blueprint for an animal. And as you know, in many complex animals like yourself, like the fruit flies I worked on as a PhD student, most of that genome isn't genes. And a lot of the genes between that fly and ourselves are conserved. But a lot of what makes us special and a lot of what makes our different cell types different is written elsewhere in the genome, in these regions we call enhancers, and they determine which genes turn on in which places and at what times in development. And because this information is distributed in this large genome, this is often happening at long range. It's able to hop over genes, etc. And this raises a question in regulation then. How is regulation both long range and yet specific? How do these sequences act of distances across up to megabases, and yet they know the correct genes to turn on and not the wrong ones? And a hypothesis that has been in the field for a long time, we have plenty of support for, is that the three-dimensional folding of the genome is one of the major guides. Unfortunately, while I was a PhD student and trying to play with some genetic experiments to disentangle this, we didn't for a long time have a lot of tools to really investigate that question very clearly. And it wasn't until the chromosome conformation capture technologies developed by Boettiger and colleagues, and then pushed to the resolution and scale by Jesse Dixon, who's here in the audience working with Bing Ren, really got us to the scales of mapping the genome at these gene regulatory sub-megabase scales, that we really saw clearly the genome is organized. So what you're looking at here, for those who haven't seen these types of maps, a representation of genome position on both sides, and the color is telling you the frequency of interaction of each piece of the sequence with any other piece of the sequence. And it's very clear from here that there is organization in the genome. It's not just a ball of spaghetti. And what was also immediately apparent from these beautiful maps is there is regulation here. There's beautiful correlation in which enhancers inside one of these boxes frequently activate genes inside the box that are less likely to activate across. And already we're starting thus to see some of that explanation of where that regulatory specificity comes from. We often will draw these features, such as these things we've called that Jesse has helped in tags here as physical structures, as boxes, which is helps schematize the picture of why one domain is separated from the other. One of the things we asked as I was starting the lab, and this is in collaboration with Bogdan, who is in the audience, and building off of some of the Murfish technologies that we had built in Zhao Wei's lab, asked, can we see these structures directly? We do this with a fairly simple approach conceptually of just walking down the genome one step at a time. We add a probe, we can find its centroid, remove it, add the next one, remove it, etc. As long as that's nicely automated, you can walk down 70 or more steps, or in some cases as many as 400 or 500. And there's beautiful work from many colleagues in the field on this. And if we average all of those little walks together, we can create another map, which is purely from microscopy. This doesn't leverage any of the high-C data on the left. And I was struck by how incredible the agreement is between these two methods at the population level. One of the nice things we can do by having the single cell structures, though, is then ask, what is the single biomolecule view of these underlying structures? And one of the things that we have been wrestling with is this observation that there's actual incredible heterogeneity at the individual cell level. And it is not just a flavor of technical noise. If you try to throw out the cells like cell 1000 here, you actually get a less good agreement here to the high C. We really need to average all of this diversity of that heterogeneity in order to nicely recapitulate the same structures we're seeing in the sequence. So this to me behaves less like a structure per se, and perhaps more like a behavior. Our analogies of folding and polymer folding, I think, have been influenced by our understanding of protein structure, and they lead us to think perhaps an overly static view. And when we have this degree of diversity, where really none of these cells have the structural features that you see in the population aggregate, it is really an emergent property, that it is maybe more behavior. So what is a behavior? Here's a behavior. I can walk through my behavior, and then I can average across my ensemble of behavior. And there we have two tags. to give a flavor of how the ensemble averaging is meaningful, but is also not an inherent structure. What I think is driving the behavior? Here's a little bit more background before I dive into the focus for today, which will be the live imaging data. Actually, I have two processes in biology that move. There's diffusion, things are moving at under thermal noise, for example. And of course, there are a lot of molecular motors. And both of these may be contributing to that. And a beautiful set of theoretical work from Leonid Murney's group, led by colleague Jeff Gutenberg, proposed that an explanation of these structures, you could get similar structures with a very simple model in which we have a floppy DNA polymer that is bound by some protein. This is going to be cohesive in a little bit, but in this theory, it's just a protein that binds DNA and it has two motor heads that walk in opposite directions. And if they remain tethered as they walk in opposite directions, they will just extrude a loop of DNA. This thing is hypothesized in this simple model to bind randomly, no sequence specificity, and to fall off and then rebind and again start extruding until it meets special sites that sit at these CAD borders, would later be CTCF, which are in the simple model merely stop sites for that motor. So this rather simple abstract polymer model, you can then create a simulation of a polymer. Here, I'll actually just show you a movie. You can see those two little motor heads walking along as they extrude the loop. I've colored the loop so that you can see it. And I hope you appreciate, as this thing moves with both those motor proteins and diffusion, there's quite a lot of heterogeneity and not a lot of block-like structure. Nonetheless, if you average this thing over time or across many independent realizations, you can get these patterns that are very reminiscent of the types of patterns one sees in the high-C data. And lots of beautiful follow-up work has provided some more of the support for this hypothesis. However, one need not invoke motor proteins to explain these structures. An alternative set of models led by many in the community, but particularly well illustrated by Mario Nicodemi's group, postulate instead a purely thermodynamic entropic mechanism that Mario called strings and binders. The scheme is somewhat simple. The polymer is decorated by different sites. The green sites are bound by the green proteins, and they will cause them to, all the green elements to stick together. The red ones are bound by the red proteins, and they cause the red elements to stick together. And in a very simple two-state model, you can get two things that on average also will look CAD-like, but in this case, invoke no motors. Slightly more complex variations of this model with 9 or 13 states integrated can provide very nice agreement to all of the different high-C data that have been produced. This model, you can in principle also capture the heterogeneity, as in this nice work from Kante, also again using this motor-free model to get an estimate of the distribution. So, what we've shown so far, I've given you this incredible heterogeneity, and I showed you the possible mechanisms that we really can't distinguish purely in the snapshot static space. One of the things I think that truly differentiates these two models is how they behave in real time. And for this, while we have postulated and used simulations to challenge our fixed cell data for a while, we have in the last several years decided it's time to try to measure these dynamics directly. And this has been led by a bold postdoc in the lab, Zhu Li, working as well with another postdoc, Lang Fu, to see if we can turn this heterogeneous structure into a measurement in time. The particular goals we'd like to address is to understand at what timescales the different 3D genome features, such as the loops, TADs, compartment structures evolve. How quickly do they change? In a snapshot, we see all these differences, but how long does it take this to get to that or that to get to here? What molecular mechanisms regulate specifically the dynamics? Some dynamical processes will have signatures in the static data, and we've been trying to measure that, but we'd like to get a better handle on those that actually regulate the speed at which these events interconvert and not just the structures that arise. And finally, can we to what extent can we measure the action of these proteins, some of these candidate molecular motors, for example, in individual live cells. To do this, Judas adopted a two-part approach. The first is a system to be able to span many different types and many different length scales relatively efficiently and still image them. And the second part is going to be getting the high spatial temporal resolution we'll need to be able to actually compare to the high spatial resolution that we can get on the fixed cell data. Here's the approach for the first part, improving genomic coverage. Building off of a lot of brilliant work, including foundational work started here by Case Murray on labeling DNA in live cells using little tandem arrays of transcription factor sites bound by fluorophores. These are nice because unlike Cas9, for example, they don't denature the DNA. You can edit them in and clone them to target specific sites. Judas incorporated two of these. There's a green one and a red one. And importantly, the red one here is encapsulated within a transposon. So having integrated this into one of our genome regions of interest, he can then mobilize the transposon and the red site will hop in different cell lines to different positions. With a single assay, we actually get many hundreds of unique target loci we can then study with live imaging. And this has been a real bottleneck in the field. Many of the exciting papers that we're starting to learn kinetics from study just one locus because it takes a lot of effort to get the first color pair. Then we hop them randomly, so we gotta know where they are. And here we borrowed a cute trick inspired from work from Mike Elowitz's group. Judas incorporated a little T7 promoter in the element that hops, facing outwards. And you can induce what they call the zombie reaction, an in situ transcription of the RNA. It transcribes into the neighboring DNA. And with super shallow sequencing, you get these incredibly clear signals of exactly where it's integrated, which has been really nice. Using the Sleeping Beauty transposon, we're able to get a nice sort of log linear scale of enriching for local events. So we have a lot of proximal jumps as well as distal jumps. And thus, we're going to span the genomic scales, again, in a single experiment. So this first experiment of mobilization produced about 750 different cell lines. They span all different genomic length scales. They land in all different chromatin types. And instead of having what those little pluses show you, or all 9 or something of the lines that have been published that we could find in all the literature, we have hundreds to choose from. Jude then chose 11 of those 700 to study more deeply with imaging, and he's chosen these to span the scales of the different structural features of the genome. So they're as close as 5 kilobases and moving roughly logarithmically up to nearly the whole chromosome scale at 73 megabases. And I'm going to use this little color scheme on the bottom throughout. Hopefully I will pop it up to with more labels. So that's on genome coverage. What about spatial and temporal resolution? So to benchmark things properly, because imaging inside the moving nucleus is challenging, I'm going to show you where we are as a field. And these are works that I think really do represent the state of the art. They're the ones we've drawn on the most ourselves in trying to build art tools. And in contrast to what you will get, or what you will see in some of Pavlov's beautiful single molecule traces from in vitro assays, we can note a few things. First, there are 3 different regions here, actually from 3 different papers. It's not obvious if I were to scramble the names, which is which. All of them do these relatively large jumps somewhere. And by large, I mean in a single frame, the jump is of order the dynamic range of the motion, which makes interpretation a little more challenging. Is that fast movement and we just miss what happens? Was that measurement error? Can we tell the difference when we see that jump in one frame? There are also periods where it looks like nothing changes. Does that mean we missed some movement or it stays static? And to illustrate the challenge faced by sparse data, I have simulated here 5 very different processes. And if I shuffle the labels, you can tell there's still 5 very different processes. But if I downsample those, it's not so obvious there are 5 different processes. I think having high temporal resolution is very important to the finer scale interpretation, though there's some very beautiful interpretation and some very creative use of statistical methods in works. Yeah.
Is it strictly necessary that you increase the time resolution? Can you also look at the PSF, the shape of the spot, and distinguish between, say, motion blur and
100%. Keep that in mind, because we're going to come back to that, because it's one of the things I think that also needs to change. That the size of the label domain does affect the PSF, and the existing work is both at 20-second intervals, but does also have a spatial limit from how punctile the PSF is. And we'll talk about that in 2 slides. I'm going to first show you where we've gotten to, and then I will highlight a few of the technical innovations to get there, to not make this too much of a detailed microscopy talk, so we have more time for the biology. So here, after 3 years or something of trying different improvements, you'll see on the left are Jude's latest data, and we're going to compare to those prior state-of-the-art examples on the right. And here is showing in a 30-minute window. The colors here represent 3 different insertions. We've chosen 3 of Jude's insertions that roughly match the scales of some of the published work, just to compare a few things here at once. And you can pick one of these regions and zoom in. We can keep on zooming in, and hopefully we appreciate the temporal resolution is better. You can see things move. Also, we have at least a bound on the spatial resolution. If our spatial resolution, if this was 200 nanometers of noise, we wouldn't expect the independent points seen in sequence to be as close together. So we can put some bound on the spatial resolution. Finally, we note that between adjacent points here are 20 to 30 seconds. Is that noise or measurement? We at least find plenty of examples where 2 individual loci do move 500 nanometers within 20 seconds. And because they move there in progressive steps, we think this is real motion, not just noise. Let's just put in some scale bars.
Yeah. Again. So is this, in which frame of reference are you measuring this? In the frame of reference of the nucleus?
Very important. So because we labeled the 2 points, everything is wrapped in the distance between the 2. So it's in the frame of reference of the chromosome itself, which is a nice way to decouple trying to measure what is the vibration of the slide, what is the motion of the cell, and what is the motion of the chromosome. Okay, a few improvements I think that help us get there. A lot of them are actually innovations we have shamelessly stolen and compiled from the community. So for example, there's some the use of these Janelia fluor dyes become very standard in single molecule imaging, actually not used in these current yet state-of-the-art studies, and really do help you fluorophore a lot. This TI Stay Gold has been very nice. Using higher sensitivity detectors also improves things. And together with this, we can get much less photo bleaching of our sample. We can also treat it with less laser, and that we can get much better time resolution. Yeah. Yeah, they do. So we're doing fast, short Z-stacks. Yeah, which is a fun place to optimize that I also think some of the existing studies, I think we could optimize more, but I think they optimize even less. Some take quite long Z-stacks where there's plenty of motion blur happening, and it wouldn't help to do it in 100 nanometer steps. So there's some fun trade-offs there too. We also made the probe smaller to address this point spread function question that Pallav said. So a lot of the early systems, in part because detectors were less efficient and because fluorophores were less efficient, use large arrays, 10 kb, 15 kb, which we know from the ORCID imaging that I introduced at the very beginning from these DNA traces is not a point source. It has some finite size, and we can use the data to calibrate a simulation here on the physical size. And there's some difference between thus where the center of mass of that thing is and the locus, which introduces some uncertainty into a measurement. Naturally, if I take the same thing, but I use a much smaller label, this is actually in simulation taking the exact same domain, just a 2 kb element, there's less uncertainty. There's less variation in my point spread function size, and I can make a better fit. This, I think, also helps to do some of the frame-to-frame jumpiness. Finally, a lot of our other high throughput fixed cell imaging had already pushed us into the realm of building the petabyte storage arrays. We take advantage of this infrastructure to also do the high throughput live imaging data. So Jude has generated a rich data set in more ways than one. It's relatively expensive still to store these even on in-house arrays, but a lot cheaper than it would be still in the cloud. And we have been able by a number of orders of magnitude to exceed the measurements that are available from prior studies, which will be useful in some of the statistical analyses we're going to jump into. One more tidbit before we jump into the biology. Can we validate our imaging system by comparing to orthogonal data sets? It's one of the things I really enjoyed about doing the MERFISH work. We compare RNA sequencing or the chromatin tracing. We can compare to the Hi-C. We'll do another comparison to the Hi-C. So you have Hi-C here on the x-axis, and we have the live cell frequency at which any two elements are within 50 nanometers on the y-axis there. We have a quite impressive correlation. This is actually the same quality of correlation we have between the live imaging replicates. If we correlate that to our fixed cells, where now they're both distances are in nanometers, we also get a pretty good agreement. We have chosen here to use the unedited fixed cells. We don't see any sign that the labels themselves have substantially perturbed the structure either. So a little bit of validation. Now we got into the, what can we measure about the structure of the genome? We've been interested in these kinetic properties. In particular, if I have two elements and they're at some current distance now, as we may have just seen them in one of those fixed cell images, how long do I need to wait for them to find each other? We call that the search process. When they find each other, how long do they stay in proximity or in contact? We're going to call that the contact duration. And how often do they do these things? We're going to call that the passage frequency. Looking across our data set of what we see here at the bottom, from the 5 KB scale up to the megabase, multi-megabase scale, at all of these, we see these little passage events. And in all these cases, this happens in multiple frames, so that giving us a little more confidence, we are observing real motions and not, again, measurement error. From this, we find a couple very exciting patterns. I'll start off with the search time. This is the amount of time it takes an element separated at first by its average distance to close the gap. And a common metric used in diffusion analysis and other polymer physics, but also gives us a metric of, for example, the enhancer promoter in that TAD, how long they should take each other to find. And I'm showing you first the data of all of these cells in which Jude has also removed cohesin with a dead rod. Because in this background, it has this beautiful, simple symmetry. You can see a basically linear relationship there on the log-log plot, which the physicists in the room will know means a power law. And curiously, the scaling of that power law is not what we expect for a free polymer. This is our common dynamic model. That's the Rouse polymer. But it is precisely what has recently been computed by Ham et al. For a dense, unknotted polymer, moving purely by diffusion. So without cohesin, the motion is largely diffusive across these 5 orders of magnitude of genomic length scales. What happens in a cell that has cohesin? Here now is the data from the untreated cells or mock-treated cells. They have perfectly intact cohesin. And you can appreciate at these shorter length scales, all of the interactions are happening much faster. The search time to go from their average separation to contact is substantially less. And then there's this discontinuity across that border. And that right there is one of the stronger TAD borders across the locus. So all of the guys to the left pretty much share a TAD. And once we're doing these substantial cross-TAD differences, there's not much effect of having cohesin or not. Yeah. 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 No, I think it doesn't have a correspondence taking the snapshot images. And we'll see if in one more minute, if that is clearer for a reason. Some of the processes we can ask are, yeah, go ahead. Yeah, it's the amount of time it takes you to go from your average separation to contact. And it's a common metric. One of the classical things we do in polymer theory is to say, I'm just a floppy polymer, not packed or knotted or any of those things, but loose. And what is that amount of time? That normally scales. It's the nice sort of simple extension of the Einstein-Brownian relation, but that scales with the square of the distance between them. Which is a nice hallmark of a diffusive polymer. And it so happens if that polymer is densely packed yet unknotted, you actually get a somewhat flattening of that curve, but that you still expect these power law scaling relationships. So two things we see here are different. The scaling relationship is different as well as it's faster. And we've lost the homogeneity. There's this discontinuity at the border. I'm going to zoom in on that to give a sense of scales. For elements here separated at about 130 kilobases, this is in seconds. They're taking 20 to 30 seconds for that interaction. So at a range that's common to many of our enhancer promoters, this is a similar distance for the SOX2 enhancer and its promoter. That interaction is happening within what would have been a single frame, for example, in some of the pioneering work from Alexander and Orion Weiner and imaging SOX2. But because we have 60 frames there, you can measure that more robustly. So it's happening quite fast, and it's also happening way faster with cohesin. It's taking nearly 2 minutes without cohesin. And the effect of cohesin, how much faster you interact, is a much bigger difference for being at farther genomic scales. If you're way down here at 5 kb, the difference is between like 10 seconds and 11 seconds. It's probably not very meaningful. Okay, so with cohesin, search times are rapid. A lot of them in this enhancer promoter scale or sub-CAD1 are on the sub-30 second. And the other thing I found very surprising here is search times are weakly affected by genomic distance, which might be a nice way to build a regulatory locus. I can put more enhancers around my gene, and I haven't dramatically decreased the amount of time they take to find each other, because for whatever reason, the scaling of the search within the TAD is somewhat flat. Now I'd like to give a little intuition for how could that be. As a simple polymer with two elements linked by here, they have some length L, some time T. If I were to double the length, my traditional physics explanation would have been I got them with a square. Certainly the intuition is though they take much longer than time T for being double the length. How do I do better than that? In the case with cohesin, I have length and again, L and T to start with. When I've doubled the length, I've also doubled the probability of having a cohesin there. Or if I had one, I could have two now on that same amount of chromatin. If Jeff's hypothesis is right, and they load randomly, which seems to fit with the available data, then I should be able to pull roughly twice as fast as we actually see it. In principle, the spacing won't be perfectly even, and thus it's not, I double the length and I take exactly no more time. I do take longer, but the exponent here is closer to 0.6 instead of 1 or 1.5. So I think motor-driven processes like loop extrusion give a natural explanation for how you can do this scaling, and it's easier to interpret how this goes if cohesin is actually loading randomly on chromatin. I'll give one more counter little example. If cohesin were to only load on the enhancer and pull it in, at best that would be doubling the length would double the time. And I would scale with an exponent of 1, not 0.6. I have twice as far to pull. I pull at the same speed. It takes me twice as long. And the genome is doing better than that. For twice the amount of time it's taking, twice the amount of distance, it's taking even less than twice the amount of time. What about the contact duration? How long do things stay in proximity? For this, I will remind you of one of those other models we just postulated. I've been invoking the loop extrusion just now and explaining that flat part of the curve. If we take these phase-separated models, one of the places they differ is in the kinetic regime. In particular, this model predicts that intra-TAD interactions, if they are made by adhesive interactions, they should be longer lived. We see an increased contact frequency because they're stuck to each other. And when they talk to somebody else who they don't stick to, those should be transient. So do we see a signature of that in the contact frequencies? And no, we don't. On these domains, the contact duration is typically on the second scale. There's no systematic effect on either side of the border. This first point, for example, immediately aside the border is ever so slightly longer lived for reasons we don't understand. Overall, it's pretty flat. Within the TAD, we also see this ever so slight but statistically detectable increase in dwell time with cohesin. Maybe this is the time it takes cohesin to kind of slide out of the way. But overall, the message is contact duration is really not strongly driven by distance at this TAD, which is not to mean that it doesn't happen elsewhere in the genome. Yeah. Yes, a fabulous question. We have not degraded CDCF, but only cohesin. Though I will add, we have chosen our loci distributed throughout the TAD, but none are on CDCF sites. And we do expect that had we anchored by doing simulations of the same process, if we were at the CTCF site, then we would get a longer time related to the residence of cohesin at CTCF. Where this is cohesin just walk right past them when it goes. Hope that makes sense.
You look at other regions.
I will show you one other region, but yeah, it's just these 11 points for now. Yeah.
Things together would be unloaded for the part that is similar to the don't have it.
Yeah, this is an important. I should have brought my little polymer. Some recent studies have done these at CTCF sites. As I just said, we want to get sort of a general to be different, as well as to get a general cluster across the genome. We're just one position randomly in the side of the TAD, not a CTCF anchor, and also hopping not at CTCF anchors, which means as cohesin starts extruding a loop, it reaches one of our little fluorophores and it goes right past it. So it doesn't stop at the two fluorophores. They're merely reporting on position. And we can tell that we don't see a discontinuity in the ORCA data at the insertion sites.
No, so I understand why you don't see this continuity. What I found surprising is that the dwell time is not fundamentally different with what we thought.
Yeah. I think this means cohesin is walking off through the other fun points. This is still with the 50 nanometer contact threshold that we showed with correlation to the high C, which has some effect. And my interpretation of this is within that second, cohesin is able to walk beyond that 50 nanometer distance. So I'm now holding two different points together. And these points have been able to separate by more than 50. And if we had higher resolution, we might better separate, that slight statistical difference that we're seeing.
But you're still assuming that those are fundamentally different processes. One is active dissociate, like you're pulling them apart, and the other one is diffusive.
Yeah, and I bet both are happening too. So in the with cohesin data, a lot of the time they meet each other, they probably just diffuse together. Some of the time they met each other, cohesin might have pulled them together. And when that happened, cohesin didn't stick holding them together very long. It continued on to hold some other guys together all within the second, which, yeah, to me is sufficient maybe to explain this like sub-second slight difference there, but otherwise similar. Yeah.
Just curious, what's your time resolution?
Yeah. This is a very important question. These are also quite close. I made some points about the Nyquist resolution. Others on these. These are quite close to our Nyquist resolution. We're every half second. So we are probably some of the actual residence times may be way slower, and we're estimating them too high at the half second or second. And that can also scrunch these together a little bit more.
Visible. If I understand correctly, that your phasing cohesion is like a catalyst for bringing two positions together.
Yeah.
And if you don't have it, it's purely.
Possibly. This is a fun question to debate in the field. I would say if some enhancer promoters, like those that have CTCF, we see in their population average contact frequency, they also reside way more. For these ones, and I'll come back also some more TADs and some of my favorite enhancers later. Some of our favorite enhancers in our resolution we don't see in snapshots that interaction much more frequently than we see the interaction between the neighboring sites, which is where we fit the like kiss and run flavor model. And then there's some controversy over this with the micro C and other parts, which would be fun to get into, which is maybe a good one for questions. We're doing a good bit on time. I will quickly show you another TAD. So people ask, do we do this at any other domains? So that those 11 sites only really cross one big TAD border. Here is a different TAD border, and to be a little bit different this time, Judah selected distanced matched sites. Everybody is 100 kb away or 400 kb away, but one is either across the border or one is inside the border. And once again, you can see the passage time and the search time for these things on the left that will be cross-TAD interactions. Again, logarithmic scale, they're slower. It takes longer for them to find each other. They make fewer passages, but there's no effective contact duration on either side. So some conclusions so far. Then I'll do a little bit on the single molecule imaging of the cohesin speed. I claim that cells have achieved super diffusive interactions within a TAD. That's because that scaling can't be explained by any of our diffusion-based polymer models. We need to invoke an active energy burning source like the loop extrusion. And if we actually get rid of cohesin, the thing collapses rather nicely to the unknotted diffusive polymer, which suggests there aren't other, at least in ES cells, major proteins that dominate diffusion to the same degree. In this particular TAD, where we've done this at least, the local search is, to me, fast. By fast, we mean a lot of those searches within the less than 500 kb are in the sub-minute regime with cohesin, rather than the many-minute regime, and partly decoupled from genomic distance. I think this fast search has some interesting implication that I have some bonus slides we can unpack a little bit. But it means, first, if you hit image slowly, you miss some of the action. But also, I'll try to unpack this second claim a little bit if there's time. There's more time for gene regulation to integrate the events. And I will, in a moment, invoke the analogy of how we think of transcription factor binding increasing these days also being short, reversible interactions. I think of the enhancer promoter interactions are also more precise if they're short and reversible, which I will come back to in one slide. But I wanted to show you some of our attempts to measure the speed of loop extrusion from these data. We're inspired by the beautiful single molecule in vitro imaging experiments where you can stick DNA on glass, stretch it out under flow, add cohesin, and see traces like this. And in the amount of time it takes to form that loop, estimate the speed of cohesin extrusion, which was estimated at a kilobase per second. We asked, how do we do this in vivo with our data? One challenge we have is the genome is not stretched like it was here on the glass slide under flow. It's natively crumpled. And you can see what I mean by that with some of these examples of the chromatin tracing images. What does that mean? Here's a simulation then of an extrusion process. And much of the time that cohesin is pulling on these two, it hasn't actually changed the distance between those at all because it's just pumping out the slack. And I think it's easier to see in the movie. So there we go, extruding, extruding, extruding. You see the red guy got farther away. But here also is our potential solution. If I play this again, once all the slack is extruded, if cohesin is in between, we then see a short interval of highly precessive motion. So this is what we're going to look for. We tested that first in simulations. So in simulation data, you can find these precessive motion events. If you find motion that's sufficiently precessive and you ask, was cohesin there or not, because it's a simulation, I know ground truth. Anytime it made that long precessive motion, it needed cohesin. Diffusion doesn't move in the same direction for one time. Yeah. Yeah.
generally relative to each other, right? That's why the dwell time when they're in contact different than the dwell time when they're in contact. You don't have to do.
Yeah, it might be a question of scales or at what point you dwell time of the, I think you stay in the water vicinity longer, but the amount of time you actually spend in close, we can talk about this more. We can do this simulation, by the way, under the same contact analysis. Beautiful thing about simulations, and we can show this, at least in this parameter regime, is giving a similar phenomena on contact. So we look in our imaging data for examples of these precessive traces. And here are some of these. This is just time going along the x-axis, and show you some of these graphically. That there are rare intervals where we see this quite precessive motion, and instructively, we see these almost exclusively in our cells that haven't got cohesin degraded. We got 19 or something of those. There are actually 2 somewhat precessive motions that happen in the treated cells. That might be a partial failure of the degron to entirely remove the cohesin. But there's an incredible bias towards doing precessive motion only when I have cohesin. And then we use this to ballpark the extrusion speed. We have directly here nanometers per second. That comes out about 70 nanometers per second. And we like to ballpark this to kilobases per second. To do that, we take an estimate of how much stretched chromatin is going to make up that 1,000 nanometers or that 800 nanometer starting distance. We benchmark that, for example, here against some of the beautiful cryo-EM from Claude O'Shea down the road, which is using a ballpark conversion of the 1,000 nanometers to 40 kb would give you the 2.7 kilobases per second, or across the many different traces that we see, around 2 to 3 kilobases per second. Notably, to match the in vitro 1 kb per second, you would need to assume that that 1,000 nanometer stretch was only 15 kilobases, which I think is a little bit more of a stretch, but we can start to place some bounds on those. So we're inclined to conclude that in vivo it is marching a little faster than we see in vitro, which from a short discussion with Jan Michael Peters makes sense from the existing mechanisms. In particular, it's walking along a chromatinized template. And if you walk in a swing and clamp as currently modeled, you should actually move faster down the DNA. And we can benchmark this approach again with these simulations, and we see doing that in simulations, we do a reasonable job too of estimating accurately the cohesin extrusion speed. So those are the key biological points, and I have a little philosophy here on the implications for gene expression, because we haven't in the live cells measured the gene expression, but for me, one of the take-homes has been that these interactions are still fast, short, and transient, and as I said, that looks a lot like the how we think of enhancers right now, having many short transient binding events to their transcription factors. And by having many short transient binding interactions between the enhancer and the promoter, the promoter can more reliably sense what that 3D structure is. And I'll skip this piece and say, I think that's particularly important for explaining some of our earlier biological results, which is we can see nice tags like you see on the right. When you disrupt that tag border, we can make these beautiful binary changes in gene expression. But if you ask on a cell-by-cell basis what the difference in contact frequency is, we often have 1.5-fold, 2-fold difference in contact frequency, yielding like a 10-fold difference in gene expression. It's not like cells on this side of the border, enhancers on this side of the border, never see those. They actually see it quite a lot, just half as much, but somehow you got to turn that half as much into a reliable readout that happens on a per cell basis. And if you can average over many contact events, you can tell that you're.
I guess
Yeah.
from the point that's required, this has A compartment, B compartment.
Yeah.
In terms of
Good question. So everything I showed you today are all in A compartment. Our long jumps hop over some B intervals, but we didn't select any loci too that are anchored in B. And I do think those A-B border differences would be really exciting. We might more see that sign that we expect the phase separation or the different contact dwell times, etc. there, which would be really cool to look at next. Cool. Okay, I'll summarize here. This was one more illustration of accounting for this tremendous cellular variability in the snapshots and motivating that if you can move as fast as we observe things to move in the embryonic stem cells, there's hope that on transcriptional timescales, each of these individual cells can start to sense that tad border or know that that boundary is there. I'll summarize here. I had a mini summary in between that I already pointed out about the effects of not diffusion dominated, and thank the lab members. Happy for a little more discussion.
Fantastic. I think we have time for a few questions at least.
Yeah. Yes.
Very cool. So I had a question. I mean, so there's all this debate about unidirectional versus
Yeah.
bidirectional.
Yeah.
So I would kind of assume that if the loop is originating in between your two probe sites,
Yeah.
you're gonna see basically two probes coming together because of the process.
Yeah.
Bidirectional.
Yeah.
And then if it's outside probe sites,
Yeah.
you wouldn't actually see it until it's past one of them, and then it would be directional. Like this accelerated motion.
Yeah.
Can you actually see if there's different behaviors of the accelerated motion? Like, are there ones that seem to be more accelerated or like?
Yeah, I think this is a cool question. If I understand it correctly, I think to do this well, we would need to have a third point as a separate chromosomal reference frame. Because in correcting for the chromosomal reference frame, like what is not the cell moving, we've reduced the problem to the distance between the two. But then I can't tell, is this guy moving processively or is that guy moving processively? I cut that out of the system. Yeah. Wait,
Okay, I'll start.
Okay.
And it's a very interesting, exciting system. I was wondering, just in considering the original data.
Yeah.
About the repeating.
Yeah.
Right?
Yeah.
And you measure that being completely wiped out, right?
Yep.
That's one state.
Yeah.
However, when you remove the repack, and then that's a dynamic process. Right?
Yeah.
The cohesion's really looking for their positions. However, if you measure that, you kind of a control, right? Don't have cohesion at all, or it's everything's already set up. Are you measuring like the data was presented? It's in what state? Is it-
Yeah, so we measure the data after, we measure it, I think, 1 to 2 hours after the addition of the D tag. So we believe we were measuring a system where ideally the cohesion is all fallen off and we are into a diffusing polymer, as opposed to the transition where cohesion is unloading, which would be cool. But I don't know.
The other direction is true as well. When now.
I'd really be excited to do the load in, yeah, which some of the experts, you might tell us more. We are having trouble with the, I think the D tag is not very reversible, which is what we have here in all the cell lines. That's a little frustrating. But I don't know, maybe there are tricks to doing that or to switch to the AID or other ways. I'd love to load the cohesion back on from an anything. I mean, a fabulous experiment, but no, we haven't figured out to do that yet. There's another one, yeah. If you there, then here.
Can I ask a more general?
Yeah.
Part of the or so for example, within intron cat.
Yes.
So you have a metric to measure that or you have a general feeling how much?
Yeah, there's a few ways you could try to, so we can say how quickly do how long do I need to wait for a 200 nanometer change? That we have a decent handle on. That's on the time scales of, if they're within tags, on the seconds, tens of seconds to the,
Direct infusion, like how much? Do you have a more more?
you might ask like how long, if I have two different 3D structures, how long do I need to wait for this one to look like that one? And for there, I need to measure more than 2 points, which was the original idea of why we tried to improve the spatial temporal resolution so that we could track more than 2 points, but even more complicated. More. Let's take one in the back and then here, yeah.
What was your effect if For example, if you remove waffle inside of it,
Yeah,
if you made up one of these waffle, I think.
yeah,
Saying that actually, at least like in both level, like the internal touch area becoming more depleted.
yeah, yeah. No, I think it's a fabulous question. What happens when you deplete waffle? From the static data already, and do you expect to some of as the loops go longer, maybe some of the local interdistances get bigger? I expect the speed should possibly go down a lot because the cohesin is arrested at its end sites and isn't pulling in the middle. And we attribute from the data and the simulations that all the kinetics are really due to the moving, the walking cohesin. So I think that would be a really exciting move to look at. Yeah.
Okay, thank you. And just regarding the derivation, regarding condensation versus.
Yeah, yes, yeah.
At the same time, can you elaborate more, like how does it go along with the kind of other observations in the field, the condensates and so on?
Yeah. Broadly, I think for explaining these pads, it is at least a more simple parsimonious explanation of these are formed by cohesin preferentially making contacts between these two. So I think this is a border that if we also look in the snapshot aggregate data or in the ORCA, does also disappear upon cohesin depletion, that we would expect not to see that residence time difference. Where we previously looked at polycomb domains as finite pieces of DNA. These also, they're neat little tags. They are happily there without cohesin as well. Our model and the model in the field for how that's happening is the sticky interactions between the polycombs, and thus we would expect the interdomain dwell times to be longer. We haven't measured this yet, but that's my prediction, and I would love to test that.
All right. In the interest of time, thank you so much for a fantastic talk. We hope that we can reel you in and actively diffuse you back here again soon. This has been super illuminating. Join me in thanking Alistair once again.
All right, thank you everyone for watching.